Hey. Mhm. Mhm. Yeah. It's going to be Zoom. Yeah. Yeah.
We're just gonna.
Yeah. Oh, good. Yeah, I don't have a link on there, but I'll post the link in this afternoon. Okay, let's see here. What do we got? Zoom. Share the screen. Share. All right, let's see here. Let's get started. Okay, everybody. Congratulations. You made it to the last lecture of PIS 220. We are going to have our final exam. This is, remember, non-cumulative. We've got endocrine, reproduction, brain and motor systems. It's going to be Wednesday, May 7th, 9 a.m. You can show up at 8 if you want to. Nobody will be there. Okay, it'll be 60 minutes, 50 multiple choice questions, just like last time. This is where you're going, either here or next door, depending on your last name. Okay, I'll do a review session on Zoom, okay, from noon to 2 p.m. On Monday, I'll post a Zoom link, probably today. On Brightspace, you have a practice exam from me. That was my 2015 exam. There's some really hard questions on it because I was really a gunner back then. I was going to be really hard ones, so don't get too worried if you can't answer all of them. But it's there. We also have Professor Bearcat. It's got her exam review guide and her lecture notes, which are really detailed, fantastic notes. And that is really quite well aligned with the content that we covered. So that will be useful to look at. I'll put up my own. I'm going to write a sort of more concise study guide that I'll put up hopefully by the end of today. Okay. Any questions on those logistical issues? Okay, well, then let's begin. So we're going to look at brain and motor systems. The brain is designed to create motion, right? It's to take in information from the environment. What is the trajectory of that football? Where am I going to have to move to run to catch it, right? And then go and execute that movement, coordinate the activity of your entire body, interact with the external world, and succeed in whatever task you are trying to accomplish, right? And it could be diverse things. It could be sports. It could be like something cognitive, something creative, something writing, anything that you want to do. Some social interaction, right? This is all going to be embedded through the integration of your central nervous system talking to your body through the peripheral nervous system and taking actions, right? And we've seen this slide before. We're taking in sensory input. We're considering it. We're doing information processing on it through specialized neural circuits. We're then sending that information back out to our muscles, cause changing contraction of our skeletal muscle, changing the body, changing the tongue, right? And then seeing what happens outside in the world, and of course, we're going to take feedback. What was the outcome of our actions? Much of the brain is designed to, one, make predictions about what, if you take this action, what would happen. Most of your time thinking, I'd say, sort of stewing over things in your head, thinking about stuff, is actually just making predictions about what's going to take place in the future, right? If I do this, what will happen, right? And then the other half of your brain is pretty much thinking about what just happened and how did I influence that outcome, right? How will I change my behavior in the future in order to have a different outcome? And that's all going to be controlled here by this system, this nervous system. So we're going to sort of briefly go into peripheral nervous system. There's just a few sort of things. So we can divide the peripheral nervous system into afferent and efferent neurons. What is afferent and efferent? Okay, it's just like an afferent and efferent arterial. Afferent is something that's bringing something into an area. Okay, I also could call it afferent. I call it afferent, whatever. Okay, afferent starts with an A though. Okay, it's bringing in information. It's bringing in blood to the glomerulus of the kidney. Okay, it's bringing in information from the external world, sensory information. Then we have efferent neurons. What is sending information out? Okay, and we can go in two different directions with that. We can go to the motor system, so that's the skeletal muscles that are going to control the positioning of our bones, allow us to move through the world, and also to the autonomic nervous system. That is going to control the parts of our body that are not muscles. But there's lots of stuff that we need to control. We need to control our lungs. We need to control our heart. We need to control like different hormones being released. We need to control like digestive juices going out. We need to get signals back from there in order to understand what we just ate and change our behavior for that. So this autonomic nervous system is going to sort of control that introspective aspect of interacting with the body. Okay, so this is going to be basically looking at how by the motor system will control how we're going to interact with the world through positioning of our bones by muscle contraction, and then our internal world is going to be sort of related to or interfaced with by this autonomic nervous system. We can divide the autonomic nervous system into two opposing pathways. Okay, we have our rest and digest pathway and our fight or flee pathway. That's called the parasympathetic division and the sympathetic division. Okay, now those are going to come out by distinct pathways. We're going to interface by distinct pathways. Okay, the parasympathetic division goes out via cranial nerves, okay, that come out of the brainstem, okay, down here. Okay, and they are individual nerves, like bundles of neurons that will come out and innervate different parts of your body. A lot of stuff in the face, your eyeballs, okay, your muscles in the face, but also you're going to have cranial nerves that go down to your lungs. You're going to have the vagus nerve, which is a nerve that's going to wander all throughout your body, okay, and touch many of your different organs in order to cause a coordinated response to a stimulus that makes you want to rest and digest, to slow things down. Okay, so all of those actions are going to be coordinated together to try to bring a change in tone, a change in your overall energy and focus into resting, right? So what do? We're going to reduce the amount of information coming in. We're going to constrict the pupil of our eye. We're just not paying attention anymore. We're relaxing. Eye pupil constricts, okay? We might start salivating. We're going to reduce the size of our bronchi in our lungs, slow down the heart. We're going to start releasing digestive juices, get the pancreas going, okay, gallbladder, all those things that you need in order to acquire nutrients from the environment that you've stored up. Okay, you can also release the bladder, get an erection. All those things are related to the parasympathetic nervous system. So relaxation. Okay. On the other hand, if you want to engage more fully in the environment and pay attention to what's happening around you, you are going to tend to shift up that tone. And that is going to be accomplished via the sympathetic nervous system. The sympathetic nervous system does not go out via cranial nerves. It talks to the body via the sympathetic ganglion through the spinal cord. So we have along the spinal cord and these different sort of sections, the cervical up here in your neck, thoracic in the middle of the back, the lumbar, the lower back, and the sacral, basically your butt. Okay. All of those, at each vertebra of your neuron, there's going to be these dorsal root ganglions that come off the back, sympathetic ganglions that are full of nerve bundles that are going to go out and have bidirectional communication with different parts of the body. Okay. The action of all of those is going to be to increase your activity level, increase your readiness to take action and movement, increase your attention. Okay. So what happens when we're trying to increase our attention? You're talking to somebody you really like, you're really interested in. What's going to happen? Your pupils are going to dilate. You're taking in more information. You're listening more closely. Okay. You're not going to salivate as much. You're going to relax bronchi in the lungs. Your lungs get bigger. You can accelerate your heartbeat. Right. All of your gut starts getting inhibited. Okay. Whereas your adrenal gland starts to get stimulated. Get some norepinephrine going. Get some acetylcholine getting released. Okay. And all of those things are going to be coordinated by the sympathetic nervous system. So these are two divisions, okay? And you can imagine, like, you might want to, the reason it's organized this way is because you want to have a coordinated action across multiple organs and across the whole body. You're trying to push it one way or the other way. Okay, so that's done. Peripheral nervous system done. How about central nervous system? We looked at this before. Central nervous system is the brain stem, and the spinal cord. The brain is mostly the cerebral cortex. Cerebral cortex, 80% of the weight of the brain, only about half the neurons though. Okay. And this is the part of the brain that truly has expanded dramatically in mammals and gives rise to our intelligence, right? It has a lot of specific parts of the cortex are going to process specific types of information. Okay, it could be sensory information, hearing, vision, audition, or I guess that's hearing. Okay. Motor, there's other areas that would be planning and execution of motor control, and there will be association areas that are going to be interweaving between those things where you're going to have to combine information from multiple sources, deliberate, then take action, right? All of our language processing is going to be like the understanding of language is complex, right? We have to take in like sound, vibrations, okay? And then we have to then map that to some words that have some semantic representation, and the order of the words matters, right? It's really complicated. So there's a lot of different brain regions that are going to be involved in that processing process. And of course, articulating our thoughts, translating what's going on, this pattern of neural activity into something that can transmit a knowledge to someone else through like patterns of motor control. Absolutely crazy. How do all these things fit together? These are all part of complex, interwoven brain systems. Now, looking just from the pure anatomical, simplest anatomical perspective, let's just take a slice through the brain. This is a coronal section. I'm not going to bother you with like learning all the different angles of slices and stuff, nomenclature. If you're going to do graduate work, sure. Okay, you don't really need to know that for this course. Okay, the coronal section through the brain. What do we have? We've got this outer 2 millimeters of the cortex. That is the gray matter. All of the neurons are there. What is all this other stuff? The white matter. That is just the wires that are connecting the neurons. Okay? Those are myelinated axons, okay, that are moving information from one part of the brain, one specialized region. I just processed some auditory region. I'm trying to find some auditory pattern. I need to go make sure my visual system has knows about that, so I can put those two things together into an actual percept of there is a bird chirping right over there, and I want to catch it or something, right? So you need to inform. You have to be sending this information, distributing it across all the other parts of the brain, and as you might imagine, that takes a lot of wires, and that is a key constraint in understanding how that brain is organized. Okay, you can unfold this surface of the cortex. Okay. And it will be about this big, okay? Poker table size. Okay? So in order to cut, and why, so the huge surface area, the expansion of the surface area of the cortex is what has caused the increase in our intelligence. So the bigger the body, the more surface area you have to have. So whales have got a really big brain because they just have a lot more muscles and stuff to move. Okay. But on top of that, if you look at the ratio of the size of the brain, size of the cortex specifically, to the size of the body, humans have by far the greatest encephalization, the greatest ratio of that. Okay, so our brains are disproportionately larger. Our cortex is disproportionately larger, and that is probably because language developed and allowed a positive feedback loop to occur where we could socially interact, build technologies, communicate with one another. The people that communicated better were able to organize societies better and outcompete everyone else. Our brains got bigger and bigger and bigger. They got so big, it's really hard to deliver a baby, right? So we're basically at the limit of how big you can create a brain and still get it out of the vaginal canal. So in order to continue to create and expand that surface area, because the expansion of that surface area creates different areas where we can process new information. We can identify patterns of the patterns. I want to understand the patterns of these sets of areas together, combine them. Well, I need a new brain area that's going to take in information from there and look for patterns in that to process that information. So the more complex the thoughts, the more brain regions are going to be integrated, and the more expansion of those brain regions we're going to need to have in the brain. So as they get bigger, as that surface area gets bigger, we got to start crumpling it up so that we can fit in the same size space. And we also, if we crumple it up, the wires don't have to go as far, right? If I have to send a wire from here to here, it's going to take a lot longer time than if I would fold a piece of paper together, right? Now my wire just has to go here to here, much closer. So this is an evolutionary adaptation. The wrinkling of the cortex is an evolutionary adaptation that enhances the ability of us to communicate and also allows an expansion of our cognitive abilities. Here is a structural MRI on the left of the cortex, the brain, similar slide plane of sectioning here. And on the right, we have a stain that stains the neuron cell bodies in blue, does not stain the myelin. Okay. So we can divide all of these brain regions into general regions that share some aspects of neural information processing. Okay. There's some specialization between left and right brain. It's not that as big as the media might have you believe. We won't really talk about it too much, but here we have an example of different brain regions, okay? And how do we figure out what is the function of each brain region? So there's a lot of distributed processing, but each brain region has its own special thing that it does really well, that's really important to doing. And we can identify the function of those brain regions through, how would we do it? Usually it's lesions, okay? A lesion like somebody had a stroke. Strokes can happen anywhere in the brain. A stroke is where the blood supply gets cut off or the blood starts bleeding out in a particular area that tends to cause the removal of oxygen in the area, causes neurons to die. The neurons that die start dumping out all their glutamate, spilling it all over. The glutamate lands on all the glutamate receptors on the neighboring neurons, and they get really excited, and they can burn themselves out too. So there's a focal area of a stroke where you have the actual damage that took place from the oxygen deprivation, and then a penumbra, an umbrella around it, where you get further damage from the glutamate, uncontrolled glutamate release. Okay, so, you just clinically, people look and say, oh, this person lost their ability to talk, but they can still understand what I'm saying, okay? That implies that the production of speech is in a distinct region of the brain than the understanding of speech. Okay. People that discovered these differences, Wernicke and Broca. Okay. So the area of the brain, and then after death, they dissect the brain and say, oh, what part was screwed up? Oh, this person was mute. Okay. Well, if they're mute, they can understand, but they can't talk. Turns out they have a lesion, stroke, or some damage to that part of the brain, Broca's area. If they can sort of make sounds and vocalize stuff, but can't understand what you're saying, it's Wernicke's area. Now, where's my thing? Okay, so there's many other brain regions here. They're all organized into 4 different lobes of the cortex. Okay. Then we have the cerebellum, and then we have this midbrain. We've got some stuff buried underneath here. Okay. And then this midbrain brainstem. Okay, they all do different stuff. The closer you get to the brainstem, the more evolutionarily conserved that information is, right? So you might talk about the lizard brain or something, right? If you're on some podcast, oh, my lizard brain was so crazy. Okay, this is where this brainstem area, the midbrain region, that's where things like heartbeat is controlled, things that introspection is taking place, the visceral stuff, anything that's interfacing with your body that that lizard or any vertebrate really has, right? We've all got to have those basic housekeeping functions of a brain to keep our body going, right? Now, in the cortex, though, we've got 4 different lobes, okay? And they're sort of divided into the temporal lobe. This is on the sides, these little wings near our ears. That is really a lot of auditory processing or hearing. The back end of the brain is the occipital lobe, which processes visual information, which is kind of crazy, stupidest design you could ever imagine. Your eyeballs have to have retina. You've got your retina. It's going to send information out of your optic nerve. It's going to go to your thalamus here in the middle, and then it's going to go all the way to the back of the brain, where then visual information is going to start getting processed. You're going to start recognizing, darks and lights and shapes, and build that up into eventually perception across multiple brain regions and the perceptions of faces. Okay. Then here on the top back is the parietal lobe. This is still sensory stuff. So sense of touch, somatosensation, the sense of the body and touch, as well as integration, multisensory integration. If I want to understand not just what's touching my fingers, but I want to look and combine that information with what is landing on my fingers, right? To actually understand what is the object I'm holding. Okay, then I'm going to want to have some sort of area that's going to combine information from my somatosensory cortex, combine information from my visual cortex, right? So those association areas are also found in the parietal lobe. Okay. Then adjacent down this central sulcus, which is this little valley between the parietal and frontal lobes, we have the motor cortex, primary motor cortex. And there's multiple regions of motor cortex, primary, secondary motor cortex, accessory motor cortex, all these things. The primary motor cortex is the one that's closest to delivering the message to the spinal cord. The spinal cord then is going to receive the message on what actions to take and translate it into the pattern of electrical activity that's going to need to cause coordinated contraction of your muscles to implement what you actually wanted to do. Okay. Now, that's not the only thing. So as you move, so the sort of closest to the periphery in terms of the wiring is motor cortex. And as we move further forward, anterior, this is the front of the brain, by the way, that's the back of it. We move forward, we get into more and more sort of abstract types of brain areas which are going to be involved in symbolic representation, that are going to be involved in planning what you're doing, in rumination, the process of thinking about what you're thinking about, or the process of thinking about what you want to do. Those areas are all up there in the front. That's an area that's going to really define your personality, right? The pattern of connections there are going to influence what sort of behavioral actions that you will choose to take in the future, right? Damage to that part of the brain, but there's the case of Gage that had a railroad tie blown through his head, but he didn't die. He just knocked out the front part of his brain, totally changed his personality, still could live, still became an inveterate gambler, sex addict. Then we've got other people, right, that we have. So in like the 1940s, 1950s, we have a lot of, back then they had mental hospitals, right, where psychiatric hospitals, psychiatric wards, homeless people generally went there instead of, the streets. And there they try to develop treatments for how can we, fix these people that have the psychotic breaks or something, right? So one strategy that an enterprising doctor came to do was to invent the lobotomy. Okay? What's the lobotomy? It worked really well to prevent people still survive. They still could go about their day. What they did was he'd come in and take a knife, sedate the person, take a knife, stick it up their nose, get a hammer, bang it up in there, and then just go back and forth like this. Cutting all of the nerves, all the neuron connections here from the frontal lobe of the brain to the rest of the area. What did that do? It removed their motivation to act out. It removed their motivation to take many behaviors. Okay, so they were still around. They still looked like a human. They still could talk. Okay, but they didn't have these wild behaviors, and they didn't really have motivation to do much except sort of wander around like zombies. If you've ever seen One Flew Over the Cuckoo's Nest, classic film, there's also a book, right? Jack Nicholson, that at the end of that book, oops, I don't want to give it away. Guess what happens? Lobotomy. Okay. Well, there's also another end that I'm not giving away the real end. Okay, then we got the cerebellum down here. Cerebellum is sort of this almost like this timing metronome system that is going to interface with a lot of these other brain regions and coordinate the overall activity of those brain regions and how it relates to motor actions. So as you can see, there's different overall specializations of the brain in different regions. But what about within a single region? Is there an organization there? Of course there is, right? This thing is insanely organized. Okay. So one of the easiest places to see the organization of the brain, in particular the cortex, is within the primary and motor and primary somatosensory cortex. Remember, these are the two things right next to each other on the central sulcus here. Okay. Somatosensory, or S1, motor, primary motor, or M1. Guess what? They're touching. They're right next to one another. Why do you think that is? Because they are really important to talk to one another, right? If you want to pick something up, or if you want to, I mean, humans have got so our humans are great, right? We've got these huge brains, right? We've got language. That's probably the most distinguishing factor from other mammals and other animals, right? Can talk. The second most distinguishing factor is our precise motor control. There is no animal that has the ability to feel things and manipulate objects nearly as well as us with the tips of our fingers. Okay? We have a massive expansion of the number of neurons that process information about the tips of our fingers, and that's what allows us to craft such intricate tools, to do such interesting art, because we have the ability to discern sensory information at a much higher level on our fingertips and execute actions in response to that sensory information. In order to do so, we have to have a closed loop of information traveling back and forth, reciprocal connectivity between the sensory and motor areas, right? So if you want to try to tie a shoe with your fingers, no, it's impossible. Just pick up a match, okay? You cannot do it just by looking at it. You have to feel that feedback in a closed, very tight loop of information transfer between the primary motor and sensory cortex in order to execute those actions, those fine motor actions. Now, within those areas, there's what we call a somatotopic map. And this is a common organizing principle of many cortical areas, particularly sensory motor areas, but particular sensory areas. And what that means is that there is a spatial organization that places neurons that innervate or get information from particular parts of the body are next to each other. Okay? So there's a mapping of the body. So neurons that represent the tongue are all down here, or that control the tongue are down here. Neurons that control the jaw are here, face here, eyes, neck, right? All in this organization, all the way around. Okay? That is mapped to specific areas, neurons along here. Okay? And that map is reflected almost like a mirror image of it on the other side of the central sulcus in the information gathering capacity, right? So neurons that get information from our fingers and thumbs are right here. They're adjacent on the other side of the neurons that control the fingers and thumbs. Each finger has its own little map right here. Each part of the finger has an organization to it. Right? So there's a somatotopic, there's a mapping of body to the topography of the map on here. Now, what do we notice about this map? Okay, it is not scaled to the overall size of our body parts, but it is scaled to something. It is scaled to the total number of nerve endings that we have in each part of that body, right? If we want to have that fine motor control of our fingers, if we want to have that incredible ability to articulate sounds with our tongue, to be able to have languages, to be able to have the thousands of diverse languages across our different cultures in the world, that all requires incredible control of the tongue and mouth, incredible control of the fingers, right? So we need more neurons innervating those areas, connected to those areas, for both taking information in and for control of those areas. So we have an overrepresentation of those areas in the cortical surface, in the topographic map. If you were to, take an even distribution of nerve endings to our body, we'd have to scale up a certain size. Our hands will be gigantic. Our lips and mouth will be gigantic relative to the rest of our body. Okay. This also will reflect into how finely we can discriminate. You can sort of test this by trying to tell how finely can you discriminate whether or not you're getting poked by one object or two objects, 2 pinpoints, right? On the tip of your finger, you can do an incredible job, less than a millimeter precision in being able to tell if it's one or two objects. On your back, you can't even tell like with an inch apart. If you poke yourself on the back, and that's because the density of nerve endings is much higher in your fingers than on your back. And that's then also reflected in the overall sort of the density of neurons within the cortex is the same. It's even across here. So if we have, so we have a change in the sort of overall size of the area that's going into a particular patch of brain region, rather than a change in the density of the neurons in the brain region itself. Any questions on cortex? Okay. Yeah.
Do they work together or they separate?
They are all everything in the brain works together. Everything in the brain works together. But in particular, I would say these two brain regions really work together. They are constantly sending messages back and forth with very short latency, right? Because they're right next, they're put next to each other so the wires can be shorter so they can have bidirectional communication much faster than if they were on occipital lobe in the front of the brain, right? They'd have to send, you're going to take, I don't know, 30, 40 milliseconds to send it that far, maybe even longer. Yeah.
Secondary.
What?
Other secondary motor cortex.
There's secondary motor cortex. What does that do? Okay, so primary motor cortex is basically planning the exact movement of your muscle that you're going to do, or your hand. The secondary motor cortex is more like planning where I want to reach. I want to decide I'm going to reach over there. Okay. And then that's going to send a signal to the motor cortex. Okay, we're going to reach over there. Motor cortex is, okay, I need to like extend my arm in that direction in order to reach over there. And then that's going to send a signal down to your spinal cord. And the spinal cord has its own circuits because your muscles have to be coordinated in a way, right? The muscles always oppose, there's always a set of muscles that oppose, each muscle has a set of another muscle that opposes it. So you have to relax one and contract the other. So to make a coordinated action, that requires a synergy of multiple muscles moving or relaxing, contracting and relaxing together. So that translation of the signal of I'm going to reach my arm like this into the actual pattern of electrical activity that has to be deployed to the muscles is accomplished through your neural circuits in your brain and in your spinal column. And the transformation of information representation over training from your primary motor cortex to your spinal column is the basis of improved skill at a sport, like tennis, ping pong, whatever, and basketball, right? If there is an action that you're trying to take and get good at a process, right? You're going to repeat that motion over and over again. You're going to have some sort of feedback. Oh, I succeeded or I didn't succeed. And if you have to think about it, you have to really think about how you're holding the racket or something. That's happening up here in your cortex. Once you're really good at something, you don't think about it anymore, but it's just automatically taking place. That's because the exact sort of pattern for exactly how to hold that racket, to interact with that object, has now been moved to the spinal column. So you can just call on that action in a more efficient way and in a way that has less variability and more precision. Other questions? Yeah.
So like the density of the neurons is the same throughout the cortex, but then the like surface area of the cortex is differently allocated for these different pieces.
Exactly. Okay, okay, we've done the cortex. Wow. Okay. So you can, systems neuroscience, we'll do it for half a semester. Okay, what about other systems? We'll just touch on, there's a bunch of other brain systems. The limbic system is the one that controls our emotional intrinsic drive and the intrinsic drives. It consists of the amygdala, the area that establishes the valence of an environment for you. What does that mean? Whether or not you're feeling good or happy or sad or scared or not, or relaxed in a particular environment. That is occurring here, this emotional state. Okay. Then we have the hippocampus. This is going to give you a map of where you are in space. And also is involved in coordinating long-term memories formation. Then we've got the thalamus. This is a gateway between your sensory and motor systems and the cortex. You can turn on or turn off. This is where you can control attention to one sort of sense or another sense by turning on or off relays in the thalamus. Okay. We've got the olfactory bulb. This is sort of an area that does a lot of olfactory processing. And sort of, this is the olfactory bulb. Like a lot of our smell processing does not happen in the cortex. It happens over here. And it takes fewer synapses to get to the amygdala and this limbic system. If you're coming from the smell pathway than from any other pathways. This is why smells often trigger the greatest, most powerful emotional responses, because they have a greater connection to the amygdala, a faster connection, a more direct connection to the amygdala. And then finally, we have hypothalamus, which is hypo, below the thalamus. This is an area of very distinct, tiny little nuclei, okay? Each nuclei has a specific function, a specific intrinsic drive for it. I don't need you to know every little name of each nuclei, but you need to know that they have specific, they map to intrinsic drives. Many people will call them the 4 Fs: fighting, fleeing, feeding, and mating. Each one of those, activity in each one of those is going to trigger a very powerful drive to take that behavior. Okay? I'll show you 2 examples. Okay? Not 4. Aggression, fighting. Okay? This is an experiment done by David Anderson at Caltech, where he has placed a light-sensitive ion channel into a particular region of the hypothalamus. Okay? Then you're just going to shine light into it through a light pipe. Okay? That will activate neurons specifically in that area and nowhere else. Okay? What will happen when we do it? When this mouse is exploring this, little area in this cage, is chilling, the light goes on, he starts to attack and fight with a little inflated rubber glove. Okay? Light goes off, he's chill. Okay? Normally, the mouse is not going to care about this. Okay? Wait for it. Wait for it. Light is on. Activity in that part of the hypothalamus goes up, ramping up. Boom, starts fighting again. What about feeding? This is like an experiment from the movie Seven or something. I don't know if you saw it. Probably it's too old for you, but there's like each sin, someone is killed for each sin. Okay. One of the sins is gluttony, right? The guy just keeps feeding this person, right? So this is a mouse completely satiated. Okay, he's totally full. Okay. But now we're in a part of the hypothalamus that controls feeding behavior. If you turn on that part of the hypothalamus, they're just going to keep eating the cheese. Okay. Turn it off. I was like, okay, well, I'm full. Okay, let me get out of here. Turn the light on. Oh man, I'm hungry again. Okay, so this is crazy, right? And this happens in your brain too. Okay, all of those controls. I'm hungry. That's this part of your brain going on. Okay, there are very rare diseases like sort of genetic defect where you can't stop feeling hungry no matter how hard, how much you eat, right? That is going to be driven by the activity, inappropriate activity in that part of the brain. So all of your intrinsic drives are there. Thirst, all these other ones. Yeah. So let's talk about the hippocampus. Hippocampus is part of the brain. Why is it called the hippocampus? It looks like a seahorse, Greek word for seahorse. Okay, if you dissected it out, it looks like this, kind of seahorsey. Yay. Okay. And there's some really cool circuitry within the hippocampus that if you take 408 with me, we'll go into great detail about. And a lot of our understanding of how long-term potentiation takes place and neuronal plasticity takes place from studies of the hippocampus and slices of the hippocampus. Because the hippocampus is essential for storing long-term memories. Damage to the hippocampus, like in the famous patient HM that probably you read about at some point, prevents that person from making new long-term memories. Every time you start a conversation with them, it's like the first time you met. Okay. And they would do that for they studied this guy for 40 years. Every day, hey, how's it going? Oh, never met you before, right? Okay, but he still remember his childhood up until the point of the injury, okay, where it was a stroke that killed off his hippocampus and entorhinal cortex. So that's one thing, storage of long-term memory in hippocampus. The second thing that's really important doing is providing your map of space and location. So it tells you what room or what environment you're in, okay? And then it also tells you where in that environment you are in. The hippocampus has got thousands and thousands of neurons. In any given environment, about 10% of the neurons are allowed to turn on. That set of 10% of those neurons that are allowed to turn on act sort of like a QR code. If that set is sort of available, that is going to allow a coordination to memories of that place across the other parts of your brain. Okay, so that's like your sort of address. And then when you're walking or moving through that environment, different sets of that 10% turn on. As I'm walking, a different set of neurons turning on in my hippocampus, depending on exactly where I am. What are those dots? That is a rat running back and forth on a track. They're recording from dozens of neurons at a time. Each dot is the action potential that is firing from one of those neurons. Each color is a different neuron. So depending on where the animal is on this track, different neurons are going to fire. By the animal's brain reading out that information, which neurons are firing, it can know where it is in the place. So place cells. That discovery got the Nobel Prize. Any questions? Okay. Let's move to motor control. Because, this is what the brain is supposed to be doing, right? And so the motor control, so muscle activity here. Okay, this is sort of images from Body World. It's kind of gruesome, but also super cool if you ever get to see it, right? I don't know how they found these bodies, plasticized, but they did it, right? So you can see the organization of the muscle system. All of that muscle cell function is going to rely on microfilaments, which are the actin-containing components of the cytoskeleton of cells. Okay, microfilament motion is going to bring out contraction, and then muscle extension is just going to be the relaxation of these microfilaments. Okay, and so our vertebrate skeletal muscles are attached to the bones by tendons, and the contraction and relaxation of those muscles is how we're going to be able to move our limbs. Okay, muscles cannot provide any force outwards. They can only pull inwards. So the organization of our muscular and skeletal system is such that there is always an opposing set of muscles in different directions, right? So you have a bicep, right? And so you can pull your arm in, and you have a tricep that you can extend it out. Okay, what will happen is if I need to pull my arm in, my bicep will contract, my tricep will relax. It's not going to provide any external force, though. It's not going to be pushing, it's just going to relax. If I need to push my arm back out, the tricep contracts, pushing my arm this way, whereas the bicep will relax. Okay, and that's true for every other muscle in the body. These are called extensor muscles and flexor muscles. Okay, so if you're going to be bringing stuff closer to your body, that's going to be a flexor, like flex the bicep, bring it in. Okay, if you want to push, if the muscle is going to push stuff away from the body when it contracts, that's an extensor. So what is controlling that system? The motor cortex is the origination point of the plan of action that is going to be taking place. Okay, so in these pyramidal neurons, they're called pyramidal spinal pyramidal tract neurons. These neurons are going to send axons down here. Okay, they're going to cross over at the decussation of pyramids. Okay, so that the left side of the body is going to control the left side of the brain will control the right side of the body. The right side of the brain will control the left side of the body. Okay, it's going to cross over. Why they did that, I don't know. It's kind of it's dumb design, but maybe it's not dumb. Maybe there's some really important reason why it's that. But anyway, it crosses over, and then it's going to hit these root nerves, okay, along the spinal cord. Different spots on your spinal cord will have different root nerves, and that's going to organize neurons going out, and these nerve bundles going out to different muscle systems as you go down the body. So if you have a spinal cord injury, then information can't pass through at some particular vertebra. That will cause a paralysis and a loss of sensation, okay, at that part of the body, that area innervates and below. Okay, because there's a spatial organization so that the lower part of the body, your toes and sort of your genitals are from the sacral part of the lowest part of your spinal column, whereas your arms are sort of up at the cervical level. So quadriplegia, the loss of 4 limbs of control, or paraplegia, the loss of 2, is going to be dependent on where a spinal column injury takes place. So what actually happens though? So we've got our action potentials come in from the motor cortex down here, the pyramidal tract. They go on, they synapse onto motor neurons, okay, in the gray matter of the spinal column. Okay. Those motor neurons are then going to go to these muscle fibers, and they will make synapses, these elaborate synapses called the neuromuscular junction, NMJ. Okay. This is where the neuron and the muscle meet. Now, each muscle fiber only gets input from one motor neuron. Only one. Each motor neuron can go to multiple muscle fibers. Okay. Now, there's 2 major ways. So you can look at that there, right? Motor neuron 1, okay, motor unit 1. It bifurcates here. It makes its neuromuscular junction here on this fiber, all these little active zones that are going to be releasing acetylcholine. And then this other collateral of the axon comes over here and it synapses on this muscle fiber. It says, however, this muscle fiber only gets input from motor unit 1. Okay, this muscle fibers, this first, third, and fourth fiber only get input from motor unit number 2. That is the specificity of mapping of the motor system to each muscle fiber. Those muscle fibers are bundled together into a muscle, right? Packaged together. And the contraction, the coordinated contraction of those fibers is what's going to allow the motion of the muscle. Now, I can move my muscle slowly with a little bit of force. I can move it quickly. I can move it strongly, right? How do I control how hard I'm contracting the muscle? Right? Well, there's 2 ways you can do it. Way number 1, vary the number of motor units that you recruit. How many muscle fibers am I telling to turn on and start contracting? And way number 2 is to fire more action potentials to try to get it to contract harder. So pull harder on those muscles. And training in like the gym or something, you can train those 2 different systems separately, right? If you're trying to push more weight over a slow amount of time, that's going to basically be involving the number and strength of the number of motor units that you can recruit. Okay, but you can also try to, if you want to have an explosive. Sorry, I'm sorry. That's really in terms of like how hard you can push the overall activity level that you're pushing. It's a little bit complicated. I think that to get stronger and stronger, right? You need to make the muscle fibers a lot stronger, right? You have to. That's an actual change in the muscle. You get bigger muscles, right? More dense muscles, greater increase in the muscle fiber sort of volume, okay? But if you want to move faster, actually what you need to do is you have to have a greater coordination of those muscles. You have to recruit the neural activity better. You have to make sure all of your motor neuron, if you want to have an explosive movement, if you want to jump the highest, you want all of your motor units to go off at the exact same time as hard as they can. And so that is a training of your actual nervous system to get stronger at that for explosive movements. Okay, so here's our 2 ways that we can do it, right? A single twitch, okay? If a single action potential comes down a motor unit, motor neuron, okay, onto a fiber, that fiber will twitch once, okay? And you get a little contraction, okay? And sometimes you can see that if you're like cramping, you can see individual fibers going off, okay? Now, we have temporal summation here. If 2 action potentials come in a sharp, closely spaced in time, that twitch will be greater. Those depolarizations and the contraction will add up on top of one another. If we get a long train, also known as a tetanus, okay, then we'll have a very strong contraction of that muscle fiber. Okay, so normally, sort of when we're at rest or you're standing around, we're not actually at rest. You have some postural control here, right? You're all holding your heads up. That means there's a muscle tone, a level of contraction that allows you to maintain your body position, which requires a continuous firing of action potentials at a particular rate. The higher the rate you fire, the more contracted you're going to be. But normally when we're holding a body position, we have to be firing at some intermediate rate all the time. That is what drives muscle tone. Okay. So There's our bicep right. We've got this bundle of muscle fibers. Each muscle fiber is basically a single cell that's been stuck together and fused in these sections called sarcomeres. Okay, so a single fiber here is called the myofibril. The sarcomere is where a fusion of during development, there was some fusion of multiple cells together that now have created one long continuous cell. Okay, and each muscle fiber, so each one of these myofibrils is arranged longitudinally here. And if we zoom in on an electron micrograph, we can see exactly how this contraction can take place. And it's really through this model of this sort of sliding filament model where the actin filaments and the myosin filaments can move past each other. And force is going to be applied by the myosin filaments onto the actin filaments. So let's zoom in here and look at what these filaments are. Okay, so we've got thin filaments. These are just the overall sort of skeleton of the cell, right? You've got actin anywhere. This is polymerized actin, right? There's G-actin, so it's in a polymerized form. F-actin just floats around. Okay, G-actin is polymerized. It's providing you the skeleton of every one of the cells in your body. And muscle fibers, it's providing these longitudinal sort of cables that can be pulled on in order to contract the fiber. Okay. What is going to pull on them? It is these myosin molecules. Myosin molecules, they look like little bean sprouts, okay? They've if you ever looked at like a baby plant, right, that's coming out of the ground. It's got a stem, and then it's got something that curls over like this, okay? So the stem here attaches to many of the other stems. You've got these bundles of maybe 300 of these filaments, these thick filaments of myosin along here that are stuck together. And then coming off of them are these little heads. Okay, the myosin heads. Now those heads have the ability to bind onto or release the actin, and they also have the ability to turn ATP into a structural configuration change of the myosin head. That burning of the ATP is what's going to provide the high energy configuration of the head, which will then allow application of force. So each one of these heads can independently activate or independently pull on the actin. The more heads that are currently pulling on it, the stronger you're contracting. So they're sort of staggered around there. Yeah, so here we go. So here's a little binding. This is sort of a diagram of how they're organized at this medial line, the M line, okay, which is halfway between each end of the sarcomere, or halfway between two sarcomeres. So we've got these 350 filaments coming in both directions. And we have an actin binding site, and we have a myosin ATPase site. That is the place where the ATP is going to be broken and allow the change in the conformational state, structural state of this myosin head. Okay, now let's look on the thin filament. Okay, so this is an actual bundle, a helix of two G-actin filaments that are wrapped around each other, and they have a binding site, okay, on them that is blocked at rest. It is blocked by this molecule called tropomyosin. Okay, it's adjacent to the myosin, okay? And it can be moved aside. You can pull that tropomyosin to expose the binding sites on the actin, or you can close it back in. Okay, that process is governed by calcium binding to the troponin. If calcium binds to troponin, okay, that is going to change the state of the troponin. It's going to slide the tropomyosin sideways, exposing the binding sites for myosin to interact with the thin filaments. So if we have ATP around and we have tropomyosin in the open state, then we'll be able to, the myosin will start pulling hard on the actin. Okay. In order for that to happen, we have to have calcium around. Okay. Usually calcium is pretty low in the cell, right? Super low in the cell. In the muscle, when we're contracting, that means we have a high calcium level. because that is going to be the signaling molecule that allows that conformational state change. So the sliding filament model works like this. We have in a fixed position, sort of organized along these medial lines, is these myosins, okay, that have heads on both sides of them, lots of heads on both sides of them. And then around them, you've got just sort of intercalated between them, you have these thin filaments of actin, these double filaments, thin double filaments of actin, okay. In the absence of calcium or ATP, it's going to look like this. They're going to be further apart. The sarcomeres will be further apart. When we increase calcium levels, increase neural activity, motor activity, that increase in motor unit activity, get some depolarization, get calcium in, we start contracting the muscle. At full contraction, we look like this. The filaments and thick filaments and thin filaments are now totally overlapping when we've pulled the sarcomeres closer together. So this is just one sort of unit here along the entire myofiber, right? You can imagine many of these working in conjunction could have a pretty big impact on changing the length of that muscle. Excuse me. Okay. What are the steps in the process? Yes, you need to know this. Okay, we've got thick filaments and thin filaments. What is going to happen? What is the process? The steps that take. Okay, if ATP is around, hopefully ATP is around. Usually ATP is around. ATP can get depleted though if you're using muscles up a lot. Okay, but if if your muscles haven't been used that much lately, you've got plenty of ATP around. Okay, the ATP will bind to the myosin head when it's in this sort of floppy low configuration state. That binding event then is going to, there will be a breaking of the ATP of a phosphate bond off to turn into ADP and phosphate. That will change the configurational state to a high energy state. Myosin flips up. Okay. If the myosin flips up, now it is available to bind the actin. Okay. But it is only going to bind the actin if calcium levels are high, because you have to have the calcium levels high to get the troponin to move the tropomyosin aside. Okay. So if we have high calcium and we have this myosin in a high energy state, it's then can bind onto and cross bridge, basically form a bridge crossing between these two filaments, bind onto it. And then the release of ADP and the other phosphate group causes the motion and translation of the myosin back to its low energy state. That is where the power comes in, this unbinding event of ADP and P from the myosin. Now we go back to this low energy configuration. And then we can either stay there or fall off if we have another ATP around. Okay. Oh, here's the diagram of how this works. The calcium, right? We've got our tropomyosin. I pretty much already explained this, right? But if we have high level of calcium in the area, it binds to our troponin. Troponin pulls the tropomyosin aside and allows the interaction and the cross bridges to form. Okay, so let's look a little bit more carefully here at the neuromuscular junction, right? So each one of these motor units comes down onto a muscle fiber. The muscle fiber is only getting info from one motor unit, okay? And then there are hundreds of little synapses that are, or big synapses really, that are all sort of clustered together at this neuromuscular junction. Here's what one of those synaptic terminals looks like, okay? It is landing on top of this muscle fiber, okay? And there's a really elaborate set of tubes and sort of plasma membranes inside and outside of the cell that are aligned with one another that are going to allow the translation of membrane voltage changes into a release of calcium from inside the cell. Okay, so what are the steps? Action potential comes down our axon, and just like in the central nervous system, it's going to come into the presynaptic terminal. Okay, and in fact, most of how we know this is how it works was figured out first in the muscle by Bernard Katz, Nobel Prize winner for figuring it out in the 50s. Okay, so Katz figured out, okay, here we go. Electrical potential comes back down here. What's going to happen? We get a depolarization of the presynaptic terminal. We get an opening of voltage-gated calcium channels. In the presynaptic terminal, calcium comes into the presynaptic terminal, causes vesicle fusion. The vesicles are full of acetylcholine. Acetylcholine comes across here. It binds to nicotinic acetylcholine receptors. They open up a channel. We get a depolarization of the postsynaptic cell. In this case, the postsynaptic cell is not a neuron. It is a muscle, but the muscle has the appropriate conductances, has the appropriate set of voltage-gated and ligand-gated channels that will allow it to depolarize. So this depolarization starts here. It's going to be the biggest here, okay? But it's going to spread laterally down the muscle. Okay, so the ions come in, they spread laterally, you get some current, some electrical depolarization that spreads. The further away you go down the muscle, the less of the size of that depolarization. So this is an excitatory postsynaptic potential, and it's a graded potential, okay? This is a graded response. It's not an action potential on the postsynaptic side. Yes?
So the muscles, they'll depolarize the calcium.
The calcium, well, let's get through the whole process here. Calcium is involved, external calcium is intimately involved in allowing the synaptic vesicles to fuse and release neurotransmitter, right? But then on this side, okay, we get a depolarization from these acetylcholine channels, okay? Sodium and basically coming in, okay? That is going to cause a depolarization that's going to spread here. And it's also good, it's not just going to spread laterally down the sides of the fiber, but there's these little tubes that go all the way through the cell and out the other side. And that's going to bring that depolarization, membrane depolarization, into adjacent to intracellular organelles, specifically the sarcoplasmic reticulum. Okay, this is just like the endoplasmic reticulum, but it's in a sarcomere, which is a muscle. You just change the name. Okay, the sarcoplasmic reticulum has got a holy crap load of calcium ATP pumps, okay, that are constantly pushing calcium, sucking the calcium out of the cytosol into this SR, okay? However, when an electrical depolarization comes down here, you get this receptor interaction through organodine receptors. It's kind of complicated, but the ultimate result is that you get voltage-gated calcium channels to open up in the sarcoplasmic reticulum. Those calcium, which is packed into the sarcoplasmic reticulum, floods into the cytosol. Right? And then that creates a transient moment in which calcium level is high in the cytosol, which creates a transient moment in which the tropomyosin is removed from the binding filaments, and in which myosin can start pulling on the actin. Okay. Now we pump all that calcium back out, concentrate it back into the SR so that we have some level of temporal control over our contractions, right? So every time we get an action potential comes down, we get this depolarization, we get a little burst, we get a little twitch of contraction through the release of this calcium. And what happens if it gets too crazy? Okay, yeah. This is all one cell. This is one cell. Great question. So remember I said if we have a whole bunch of action potential occurring together closely in time, what do we call that? That's called a tetanus. That is derived from what happens if you get tetanus, the disease. Okay, everybody here I'm sure has had a DTaP vaccine probably more than once. Okay, diphtheria, tetanus, pertussis. Okay, so so if you are infected by this bacteria, it produces a toxin, okay, that causes your inhibitory interneurons and your lower motor neurons to get inhibited. If we stop inhibiting our motor units, that's going to allow the motor units to just fire all the time. If your motor neurons are firing all the time, that means every muscle in your body starts to contract harder and harder and harder and harder and harder. And that will last until that bacteria is cleared and that toxin is cleared, which is days. You might die because you can't relax your diaphragm to breathe. Not a good way to go, folks. Okay. So, but it is survivable if you get the right muscle relaxants and stuff, but pretty good to get that shot. So that's what happens. We have too much activity, this tetanus of activity going off on all of our motor units. What about the converse? Well, botulinum toxin is an anaerobic bacteria that creates this very, very powerful toxin, which we've now co-opted for medical uses. Okay. What does that do? Remember those like molecules that are decorating the outside surface of that synaptic vesicle? Some of those are these snare proteins that need to bind onto other snare, vesicle snare, and then the target snare. These are going to bind onto each other and allow the fusion to take place. What this toxin does is it's an enzyme that chops the snare proteins. Chop, chop, chop. Okay. Now they can't interact. You can no longer have vesicle fusion. That it doesn't matter how many action potentials you get coming into that presynaptic terminal, you can't fuse your synaptic vesicles. You cannot cause a depolarization of the muscle. You cannot contract your muscle. Now that lasts a really long time because this is not just like toxin that stops working when you wash it away, like many drugs. This is a toxin that's like an enzyme that chops up all these vesicles, the vesicle proteins. So it takes a long time to remake that protein and get it into those vesicles, okay? Especially because a lot of that protein production is activity dependent, okay? So you just can't even get the signal there to do it. So a botulinum shot, so a little bit of botulinum toxin precisely placed can paralyze a local area of muscles for months at a time. So it's used for treatments for wrinkles, whatnot. And it's also used for things like back spasms, okay, where you need to relax the backs, certain back in a semi-permanent way. Okay. So let's look at our types of muscle fibers. We can classify muscle fibers into a couple different ways. You can even either call it oxidative or glycolytic fibers. That's where you're getting your ATP from, okay? Otherwise, we can call it fast twitch or slow twitch fibers, which is by how quickly it can take action potentials and turn that into a contraction. Okay, there's a limit for slow twitch. Okay, now the slow twitch fibers are more matched to the oxidative fibers. Okay, the oxidative fibers are going to use the aerobic respiration cycle to generate ATP. This is a very efficient process. It uses oxygen. Okay, you get a lot of ATP, like what, 36 ATPs out of that cycle or something. So they've got tons of mitochondria. They're full of mitochondria, rich blood supply, lots of myoglobin around, okay, to shuttle that oxygen around, okay. On the other hand, glycolytic fibers are going to use glycolysis, so the breakdown of glycogen as their primary source of ATP. So you have a much better ability to continuously supply oxygen to your muscles over hours at a time. So if you need to do an endurance exercise, you're going to be using your oxidative muscles. Okay. If you need to go faster, if you need to contract harder, you can get a sort of a burst of energy from your glycolytic muscles, glycolytic fibers, but eventually you run out of glycogen. Now you can't contract anymore. Okay. So we have a blend of these slow twitch fibers, which are generally oxidative, and fast twitch fibers, which are a blend of oxidative and glycolytic fibers that can contract more quickly, but don't last as long. You can see this in a power. This is a power curve. Okay, this is how much power one person, guess who, can put out at different durations of time. Okay, and so this is 1 second. This is like 2 hours. So in that first second of contraction, okay, let's say you're trying to do a long jump or something, or just try to push something as hard as possible one time. That is all driven by ATP that's just available, immediately available. That ATP is basically used up in a few seconds. Okay. After ATP, where do we get it? We get it from creatine phosphate. Creatine phosphate can replace ATP very rapidly. This is why the most popular supplement in athletics and the one that is actually really useful, it doesn't hurt you, is creatine. Okay. Creatine will increase your short duration power for 1 minute and below. That's at saturation by about 10 to 15%. It'll also increase your body weight by about 2 to 3% due to increased water retention. But I definitely take creatine. It's great. Now, in this medium range, okay, This is anaerobic efforts, 5 minutes, 10 minutes. This is where we're using a lot of glycogen. Those are our glycolytic fibers. So this is type 2B fibers or fast twitch, okay, that are exclusively basically using ATP and creatine type 2A. These mixed oxidative and glycolytic fibers, we're using glucose, we're burning glycogen. The glucose is the oxidative part, the glycogen is from our intracellular stores, okay, of glycogen. And then if we need to go for an hour though, we run out of glycogen. We want to keep going for an hour, we need to be only using glucose. We need to be breaking down fatty acids and proteins and turning that into ATP. That's through processes of aerobic respiration. And that's exclusively accomplished by our slow twitch fibers. So you might be able to sustain 250 watts, 300 watts here for an hour, but then you can do maybe 4 times more of that, but only for a few seconds because of different fibers get pulled online and fatigue faster or slower. Okay, so how can we actually control these muscles? We know in the motor cortex, there are sets of neurons, right, that encode basically are sort of encode a different direction that we want, say, an arm to move. Okay, and here is the neural activity of one of those neurons in 5 separate trials. When a monkey moves his arm in one of 8 different directions, you can see that in some directions, the neuron fires lots of action potentials, and other directions, where this line is, there's no action potentials. It goes quiet, gets relaxed. So within the brain, many different neurons all have particular preferences for which direction they'll get excited to and which direction they will get inhibited to. And you can sort of plot that here, okay? This is the overall level of activity based on which movement direction. Here are different neurons, different tuning characteristics. So one likes to go this way, one gets excited if you go that way. And the brain basically adds up the sort of weighted vote of all of those neurons and their activity to actually determine which direction they want to move the arm. So that's how the brain does it. We can also use computer interfaces to read out that activity, okay, in order to move robotic arms. So here is the key breakthrough here, which was actually over a decade ago now, by Andy Schwartz, but it's done in many other labs, including here at USC.
Just have to say verbally, it's not that.
So here, yes. Molecular arrays in our motor cortex, they're decoding that pattern of neural activity and mapping it to
One small. One giant fight for DCI.
So by BCI, she means a brain-computer interface, right? So this is an area where if you can get the electrodes into the brain, into the motor cortex, you can read out that activity. You can teach a neural network to map that neural activity to control of a muscle, of a robotic arm. Those systems are getting better and better over time. And of course, we also need to be able to put that activity back in. This is the goal of companies like Neuralink and Synchron and stuff like that. Now, there's other diseases of the brain. Parkinson's disease is a disease where we have dopamine neurons that die off. Dopamine is involved in that reward process, but in a particular area of dopamine neurons, the substantia nigra also controls the overall activity of the striatum, which controls muscles, okay? So when those neurons die off, we lose the ability to coordinate control of our muscles at rest, okay? In order to replace that activity that's lost from the death of those dopamine neurons in that particular brain region, there's treatments like deep brain stimulation, okay? So deep brain stimulation basically implants a electrode and a battery pack down here, okay? And it's going to drop these 2 electrodes into this little portion of the midbrain here by the globus pallidus, and then try to recreate the activity pattern, electrical activity rhythm that those dopamine neurons will be giving to this striatum in order to cure, in a sense, or at least treat tremor of Parkinson's disease. So this guy has Parkinson's disease and a deep brain stimulator. Let's bring it off. Right
Okay, you won't be able to read that, it's shaking too much. So it's almost an instantaneous reaction. I can't control this. Shake some quality cocktails with the right. The left just looks like the royal wave. My voice is gone. It's harder for me to get words out and speak. I'm completely rigid. And if I tried to stand up, I'd fall. If anyone thinks I'm faking this, I'm not. I can't control the tremors and dyskinesia. The twisting in my neck is called dystonia and is really quite uncomfortable. So as you can see, the tremors are getting much worse and I'm in quite a bit of discomfort. And I'm about to drop the remote control, which will be fun because I won't be able to turn stuff back on. So I think party time is over. Almost automatically. Power comes back on. I'm steady. So if anyone's considering DBS, I thoroughly recommend it. It is life-changing. Thanks.
This does not work for all Parkinson's patients, but it does work for a sizable fraction of them, and they're trying to now develop DBS treatments for other brain conditions. But this is a great example where neural circuits and the actual pattern of electrical activity and the appropriate pattern of electrical activity or aberrant pattern of electrical activity in your neural circuits really has a profound impact on how your body is being positioned and controlled. Normally, I would end the lecture here, but I have one last thing to talk about. Okay, which I've not talked about before because I never saw the need to do it. But, you know, when I was, but I think it's important. And, you know, my job is not just to teach this material, but also prepare you all to succeed in your professional academic lives, academic and professional lives. And, you know, I said early in the class when I started, use ChatGPT for stuff, okay? The capabilities of that tool have gotten even better in the last 2 weeks. Okay? To the point where I think we basically are at the point where we have artificial general intelligence. Ask the best models a question, it will perform better than a human expert on anything, okay? And this is a plot of the capabilities on answering PhD level science questions over the course of the last 2 years and all the different models that have come out. Okay, so, you know, ChatGPT-4 is down here, basically guessing randomly, a little bit better. In the last 2 weeks, in the last month, Gemini 2.5 Pro, O3 has come out. I've been interacting with O3. The agentic abilities of O3 to do anything are absolutely insane. In a single question, you have chain of thought reasoning, it's all embedded, and it will spit out basically a research paper that is pretty good. Okay, so I want you all to understand that this is going to completely change how education takes place, and it's going to change the workforce. Okay, and I want you to have one homework assignment after the finals and throughout the summer. Every day, log into one of these models, a frontier model, one of the best that have these capabilities, just ask it to do one thing to help you out with one thing you're worried about. Because this world is going to change. I think education is going to change and jobs are going to change pretty rapidly in the next 3 years. So just keep up with it, okay? Try to learn how to use those tools in anything that you do, and you will be able to outcompete other people that are still have their head in the sand about it, okay? That's all I got, folks. Thank you. I'll see you on Monday for the review session.